So I just progress. So we are the products of our genes, correct? Yes and no, like often is the most correct answer. We know that, for example, parental care and early life experience affect the risk to develop neuropsychiatric diseases. So obviously, our environment, including a very early one, plays important role. So this term, epigenetics, right, beyond genes, was actually coined by Sir Conrad Waddington. He was a developmental biologist, and he used this epigenetic landscape that he drew to sort of illustrate how a cell, when it starts to develop, can end up in different ways. Now we actually adopted the term epigenetic when we want to talk about a long-lasting change in phenotype and gene expression that is not due to the changes in DNA sequence. So what are the epigenetic mechanisms? DNA methylation, histone modifications, and non-coding RNAs that I'll be talking about most of the time. Why are these modifications important? Well, they actually, our genes that are nicely coiled in this DNA, they actually, these modifications affect specifically DNA and histone methylation, histone modifications. How open is the DNA to transcription so that actually genes get transcribed? You need to have euchromatin for that. If it's all in this very dense chromatin, there's not going to be, actually, it's going to be as if these genes do not exist. So non-coding RNAs actually have a little bit more complicated mechanisms, how they act, and we'll talk about it a little bit later. So how do we know that these epigenetic differences actually are due to the environment? So we have this study on monozygotic twins, and the researchers have followed these 80 pairs of monozygotic twins from ages 3 to 74, and DNA methylation and histone acetylation in their lymphocytes was nearly identical when they were 3 years old. However, when they were 50 years old or older, these markers were significantly different. I want to show you this interesting experiment in mice. Good parental care versus bad parental care. What does it do to little mice? So how do you define what is a good mouse mom? The mom that is available, right? So the model is such that you have a maternal stress induced to their offsprings by basically unpredictably separated by these researchers, taking away the mom and then seeing what happens to the pups. So here you can see, of course, you always have to have controls that has normal parenting all throughout. In the first generation of these pups that were mistreated by mom not being there, you get this male mice that showed increased anxiety, depressive-like behavior, impaired impulse control. So when they were mated with normal, risen females, they get their offsprings, and these mice actually were having normal parental care. In the second generation, there were females that had these issues. And finally, in the third generation, so you have actually males who are totally fine, totally normally brought up with normal females, good care, and you still have in the third generation, you had male mice that had these problems. So these researchers obviously concluded that the only thing that actually connects these generations is a male germline, male sperm. And then so selectively, they actually looked at the genes that we know regulate depression, anxiety, impulse control, and they found methylation in those genes that actually now are targeted by antidepressant medications such as Prozac, etc. So methylation in serotonin receptors, in corticotropin releasing factors, in monoamine oxidase, all these genes that are targets of the medications that regulate this were abnormally methylated. So this is, of course, very interesting. Why 3 generations? It doesn't happen after the 3rd generation. Does that happen in humans? Who knows? Obviously not so predictably. But you can argue that there is something in the environment that the biology thinks it's good to somehow to be remembered in order to ensure that would be the positive aspect of this otherwise negative outcome of all these problems in behavior, that sometimes the memory from the environment is kept. Now, why is, I want to show you this. This is actually from the Nature, maybe like more than 10 years ago. Then they sort of, in retrospect, they revisited the work of this guy Lamarck. At that time, it was 200 years after he published this book that ended up completely forgotten. It was only translated in English relatively recently in comparison to his contemporary Darwin, who was also, like Lamarck, interested in inheritance. It took only 74 years for Charles Darwin's On the Origin of Species to be translated to Ukrainian. So this guy actually coined the name biology, and he was the one saying, like, there is some environmental cues and clues that actually can be transmitted in the 2nd generation. Of course, he was ridiculed because there was no mechanism, while Darwin's theory about mutations, random mutations, survival of the fittest, was much more intellectually easier to grasp and to explain. So recently, there was another revisit to a Lamarckian evolution theory. And in this Frontiers of Neuroscience article, the author argued that really the development of the brain cannot be only explained by random mutations, that somehow the complexity of the brain and the diseases associated with that complexity also need to be sort of, in addition to these mutations that randomly arise, are also modified by the environment. And now that we know a little bit more of biology, he proposed here that exosomes, these little bubbles that you can see in the culture here on that cell that shows the processes, they're actually a little packages of material that can be transferred from cell to cell. And what's in that? What is stored in these packages, in these exosomes, that about 100 nanometers wide, have the simple biolipid membrane, and they have a lot of genetic material, as I mentioned, messenger RNA, DNA, and these microRNAs, and also some proteins. So what are microRNAs? So microRNAs belong to this family of non-coding RNAs. They're small, 19 to 21 nucleotide long, and basically cell can turn on the transcription of the genes in the nucleus for these microRNAs, release them in the cytoplasm, and what they do actually, they can, after a bunch of cleavage and binding, etc., bind to messenger RNA, so transcription product of a DNA, and made it completely irrelevant, destroyed, as if it doesn't exist. So if basically you have silencing of the gene on the will, the cell can silence the gene. So that's obviously epigenetic mechanism. Now, why is this interesting? Of course, if something's happening at the same time, it doesn't mean that it's actually related. We know that. But as a human, we somehow tend to think that maybe there's some correlation, right? So this was published maybe over 10 years ago, but it shows really how that from the beginning of life on this earth, about 5 billion years ago, and when you had these first organisms, these archaea and bacteria, their protein coding genome was most of the genome, 98%. And as the evolution progresses, and in particular when you have this Cambrian explosion and you have diversity of life, and you also have this huge growth in the brain size and in the brain complexity, in parallel, you also have this development of this family of non-coding RNAs. Long non-coding and many small RNA, including microRNAs that I was just mentioning about. Well, so why is this? And in parallel, the genome, maybe that's the proof in the pudding, the genome gets organized in such a way that only 1.5 is protein coding and the rest is non-coding. So it's almost like you have keyboards, but then you have a much more sophisticated mechanism to play these keyboards. So, world of RNA, there's untapped potential of non-coding RNAs for biomarkers and for drug targets. Why? Because as I said, coding transcripts are only 1.5, transcriptome is 98.5. So these are these non-coding RNAs. I just mentioned them. And if you, in opinion of some, these 1.5 protein targets are mostly undruggable. So perhaps we can or could focus to non-coding RNAs as druggable targets and as biomarkers. So what is a biomarker? So NIH, I would offer us this nice definition: characteristic that is objectively measured and evaluated as an indicator of normal biologic process, pathogenic process, or pharmacologic response to therapeutic intervention. So pretty broad, easy to fit in there, whatever you like to study. Now, in psychiatry, since I mentioned we started sort of with psychiatric symptoms, there was this lamentation that I came across. Why has it taken so long for biological psychiatry to develop clinical tests? Lack of biological gold standard definition. And you can think about it, how it nowadays diagnose really psychiatric disorders, be it schizophrenia, be it bipolar disorder. This not much has changed. We don't have biologic tests. Imagine if you would be diagnosing cardiac diseases with the tests that were available 200 years ago. But this is essentially what we're doing in psychiatric disorders. So what we did long time ago, over 10 years ago, we extracted exosomes from frozen postmortem cortex that was available to us from the samples of McLean's psychiatric brain disorders brain bank. And we were actually able to show that microRNA content in these exosomes from prefrontal cortex of schizophrenia and bipolar patients differed. It was also interesting that the bipolar disorder ones are more similar to controls, and schizophrenia was really separated. And you can argue, because if you probably know, there are many people with bipolar disorder who are fantastically creative and functional individuals, except when they have this crisis. Well, schizophrenia patients, unfortunately, have a really severe cognitive issues. So, in conclusion, we can say the genetic risk factors, environmental factors, actually in their combination result in these complex cognitive and emotional disorders. And in the center is this epigenome homeostasis. So, because these mechanisms are key regulators of genome and environment interactions, as I just showed you. So now, why is that important for Alzheimer's disease? Well, the problem with Alzheimer's disease is that the key to successful intervention is early detection. Unfortunately, nowadays, when we diagnose these patients with these biomarkers that we have currently that are accepted, so-called ATN, A for amyloid, T for tau, N for neurodegeneration, by basically sticking a needle in their back to get CSF to look at the A-beta ratio, or putting the patients into an MRI. This is already after all these molecular and cellular changes have occurred. So can we somehow actually tap into that window for causative therapies before neurodegeneration starts happening? And also, when you think about it, even if we have current therapies, in order to check them whether they're working, you have to stratify your patients. You have to use biomarkers to know who is in which phase in order to actually have some sort of a useful information at the end of the trial. So all of this is recognized very well by National Institute of Aging and Alzheimer Association, and they very wisely put together this sort of a manifest saying that we need to have, although we make a diagnosis of Alzheimer's disease postmortem, we have to have in vivo biomarkers. And the ATN classification system groups different biomarkers, imaging and biofluids, by the pathologic processes that each of these biomarker measures, accumulation of amyloid, tau. But they also recognize that this framework should exist, that there is a space to generate and test hypotheses about different pathological processes rather than accumulation of A beta or tau, and that we are encouraged to seek new biomarkers. And why is that necessary? I want to show you this case that was actually a case of my colleague, Dr. Thorstein, and at Framingham Brain Bank. And how they do it in Framingham, it's really very smart, because Thor, when he looks at the brain, he just examines the brain and doesn't know anything about clinical problems that the patient had, issues the report by basically looking at, so I'm finally showing you these neurofibrillary tangles, right? Let's see if that works. You can see this is immunohistochemistry for tau. You have this intracellular accumulation on neurofibrillary tangles made of abnormally phosphorylated tau, which is a microtubule-associated protein. And also here in the lower two panels, you have A beta antibody that actually labels not only extracellular abnormalities, these neuritic plaques that have this nice course, but also shows that vessels, often not always, but often in these cases, also have amyloid deposition known as cerebral amyloid angiopathy. That is actually now becoming very important as we have this new A beta plaque clearing drugs. They actually, some of them get to clear also the A beta in the walls of these arteries, which actually then causes some unwanted symptoms, including hemorrhages. But anyway, that's another story for another day. So Thor looked at this case and said, basically, this is a BRCA and BRCA6, which is the highest in this classification of how much tau can you have, and call this AD-associated pathologic changes, BRCA and BRCA6, that's highest. CIRAD is a system to actually measure neuritic plaques. But then the committee who was, there and just looking at the clinical issues, and this person was cognitively intact when last time it was examined, which was, I think, a couple of years before this person passed away. So how is that possible, right? Interestingly enough, that person was diagnosed with bipolar disorder. So this is anecdotal. This does not happen often at all. I don't want you to get the wrong impression. But it just points out that this nice hypothesis that you first have an accumulation of amyloid, then you get tau, then you become demented. It's a framework to think about Alzheimer's disease, but it does not happen in every case. So in the era of precision, personalized medicine, we cannot just ignore that fact. So we know that Alzheimer's disease is multifactorial disease. And then this nice review paper by De Super from 2016 actually illustrates this. So this pathogenesis, biomarkers for ADs, biological and cellular, happen maybe 20 years before the symptoms, the clinical symptoms happen. And of course, it starts, we don't know exactly when it starts, but basically we have identified several genes that actually are involved in every of the mechanisms here that this slide is pointing out, right? So there is a problem with the clearance of amyloid. There is a problem of APP metabolism. There is definitely immune functions that are changed. And you can see here now that in the center, in the cellular phase, so after the molecular phase and before the clinical phase, there is this very important neurovascular unit where you can see that neuron in connection with glial cells, and is in this environment surrounded by, I don't have the pointer here. So these astrocytes are here, there's a neuron up there, and then there's a blood vessel here. That this regulation between the inflammatory cells and the support, the problems in the support that astrocytes are giving neurons, all of that dysfunction of astrocytes affects neuronal stability, affects neuronal plasticity. So all of these processes are nicely actually captured by identification of genes that regulate them. And there are accordingly numerous trials going on, clinical trials that are addressing each of these problems. Now, the question is, could microRNA signature be a signature of early Alzheimer's disease associated cognitive decline? And why is that? This is because the microRNAs actually from the brain circulate, and periphery, of course, communicates with the brain. This microRNA could be actually in the exosomes or free with some lipoproteins. And they can actually serve as a window into what's going on in the brain. Just think about an example, right? If there's a noxious stimuli or infection from the environment, the immune system cells are going to react. They're going to get into the brain. Brain cells are going to react. Exosomes are going to be released. MicroRNA is going to be released. And potentially, we can have in the periphery a microRNA that is changed in its expression, in the amount that basically informs us about inflammation. Another microRNA informs us about synaptic dysfunction, and another one about metabolic regulation. So you have 1, 2, 3, 5, and if we could detect them in the periphery, that can serve as a biomarker of pathology to happen. Now, here I want to showcase the work that was really instrumental for us finally obtaining NIH funding for what we wanted to do. My colleague Andre Fischer at German Center for Neurodegenerative Diseases published this paper where actually he, interestingly, didn't start from Alzheimer versus control. He started with normal young adults that had slight differences in their cognitive performance, how well they are performing on certain tests, reading, and so on, and was able then to identify the microRNA signature in their blood, and then from that signature, take these microRNAs and test them in vitro and in vivo in mice to see what do they actually do. And so, to cut the long story short, okay, there's a little bit of, but anyway, that signature was basically separating control from MCI as well as conventional AD biomarker did in CSF of this patient that he had access to in Germany. So that enabled us to present this data and to design this study where we're going to take the cross-sectional sample from Alzheimer's Disease Neuroimaging Initiative, which is a large study that started in 2003 and started, of course, with a focus on Alzheimer, started on with imaging, but as it progressed several phases, they're also now very keen on biomarkers. We wanted to have a replication cohort, the one in Germany, the DEL code, and then importantly, we wanted to have what was at that time still difficult to find in ADNI and in DEL code because they're much younger than Framingham, longitudinal plasma samples that were belonging to participants that also had brains in their brain bank. And we want to be extremely selective, and thanks to Tor and the poring over these neuropath reports, to have people who have only Alzheimer's disease, not also Parkinson, frontotemporal dementia, whatever, and have controls that are true controls, right? So because you can have a brain with A beta and tau present, and persons are not showing cognitive decline, it doesn't mean that this is a control brain. This is just a person that is, because of their brain reserve, coping well with this pathology that is well underway. So how you choose your subjects is always the most important thing, that will determine your success at the end. So what we also wanted to say is it's not just take a piece of cortex and just blend it and just sequence it, but basically, and this is done, the 2 years, 2 people in my lab were actually collecting by laser capture microdissection neuronal and glial cells, all the neurons, astrocytes, and microglia. And we have submitted those cells to small RNA-seq and recently identified cell class specific microRNA that change in AD. And we are now comparing this with what we found in the blood. But most importantly, we can now introduce these microRNAs in vitro to induce pluripotent stem cells-derived neurons and glia and see their effect. Because it's not enough to have just a biomarker. If something is really related to pathogenesis process, you also want to know really, you want to define that function. So anyway, ADNI was very happy because nobody else did small RNA-seq. So we provided the first ADNI plasma microgenome analysis. This was our approach. We got from ADNI and their SOPs how to have a pooled plasma that was then actually divided and submitted with each batch. And then we had this 21 batch that was necessary to sequence all of the samples. You can see that in batch 1 and batch 7, for some reason, we had samples that we had to eliminate because they were poor quality. So this is what we ended up analyzing. And we were able then to, through very complicated, above my pay grade statistics, identify these, lot of machine learning, but I'll tell you at the end why I believe these results, right? Because so anyway, we got this 70. First of all, we out of 2,876 microRNA, we identified 2,156. But then we focused only on 300 because they had enough of representation to actually make some conclusion about them. So this is our study that we published in 2024. And most importantly, again, just these are my colleagues here from ADNI, Kwansi Ko and Andy Saiken, really instrumental in selecting these participants. And these are the 2 bioinformaticians. I look very serious, right? So, so basically, we were able to identify microRNAs that already, when you think about if you can just apply this in practice, can tremendously help. Because you think about it, you go to a primary care physician, does a simple blood test, and so because of this microRNA, I think you should also have CSF examined for A beta, maybe the PET. You would actually save a lot of money, or you can say like, oh, this, you're not likely to actually be endangered to develop AD. So this could be a potentially a very useful clinical tool. And what is really interesting is that we identify those that can actually predict conversion from MCI to AD. And also what was really helpful to know, reassuring to know that most of the identified microRNAs have been identified as a potential AD biomarker, even in the previous studies that I'm already listing here. Now, this is one of our findings. And here, if you could just, up here, these are common to both those common biomarkers, actually, I should say, pathways that are indicated that these microRNAs that we found as biomarkers are regulating. And they're common to both the transition from early MCA to AD and to late MCA to AD. And you can just look at top three. This is now obscuring your screen sharing, but it says, you have to believe me, fluid, shear, stress, and atherosclerosis, and glycan biosynthesis, and lipid and atherosclerosis. So I think N-glycan biosynthesis, which is important to this, once you have a protein, you also have to put some sugars on it. And that's, I think, it's particularly important for IgG. And that is actually considered a biomarker of cardiovascular health. Actually, I think coronary artery disease. I think so. Also, in this, actually from Andre's lab, these are a couple of papers in mouse. There is this mouse model of cardiac heart failure, where basically they showed that heart failure causes cerebral hypoperfusion in these mice. There is, of course, limited nutrient supply to brain and hypoxia. There's oxidative and ER stress, which then results in changes in histone acetylation in the DNA of these neurons. And then the genes that are linked to the cognition are reducedly expressed. So that results in the cognitive deficits. They also apply this drug, vorinostat, that actually it's a HDAC, so histone deacetylation inhibitor, and then they actually restore the cognitive function in these mice who were having heart failure. So the mouse model only basically targets the cardiomyocytes, so it's only the heart that is sick in these mice, but basically, obviously, if you have a bad heart, it's not good for your brain. Now, this is acetylation. In a follow-up paper, these mice who 3 months after the persistent heart failure had severe cognitive deficits, 6 months later, actually due to compensatory microRNA network changes, these cognitive deficits were restored. So there's obviously a hope. And now I want to that you all remember this tour de force presentation that Dawn gave a few weeks ago or months ago, showing again that life essential 8 are really important for psychological health, and that poor heart health, poor cardiovascular health results in anxiety and risk of dementia, and that essentially all of these life essential 8 that are good for heart are also basically still in AD field. Neurologists don't have much else to recommend to people, but basically just follow these good life instructions, right? But this is recognized, right? European Society of Cardiology recommended that cardiology and dementia research experts team up to identify therapeutic interventions options for managing cognitive impairment in subjects with heart failure. So as I mentioned, we have currently finished our analysis of postmortem brain cells. We are now looking into the Framingham longitudinal plasma samples of this initial set of 21 participants. So once we identify the candidates that we believe, we would like to go back to a much larger sample, offspring cohort, where we identified this controls MCI and AD, and we have their samples available from multiple exams. We would like to put to the test our microRNA biomarker candidates, and considering that this is Framingham heart study, and then we also have phenomenally densely phenotyped individuals when it comes to cardiovascular health, I think we can safely devise hypothesis that we will also find biomarkers of heart health as well as brain health. And I just want to leave you here with this, that microRNAs are not the whole story. We were fortunate enough to actually got another round of funding for long non-coding RNAs, and they are particularly interesting because they are cell class specifics. And when it comes to Alzheimer's disease and related dementia, that they have specific changes in neurons and in glial cells, this is a very useful feature. However, these are much less understood and known non-coding RNAs, but we again have identified at Framingham cases of AD, FTD, AD plus FTD, again, very precisely curated in terms of neuropathology, and we also have their plasma samples. We know that they are there. We have just few rounds of questioning by executive committee before I think we can ensure those as well. And here I really want to end by thanking my colleagues. Without them, I would be nowhere. And so, but it's great to be able to work with this super dedicated and smart people, and also be able to be here with you at least once a week. Thank you.